All right, so the second part says interpret image to the right. So you study this image, and it kind of shows you what the restriction enzyme does. So the restriction enzyme effectively cut off part of this section of DNA, colored in red, and then you have this blue section that's also been cut by restriction enzymes. It looks like the restriction enzymes are shown as these black dots here. All right, so restriction enzymes have cut DNA from some source here, and then from a different source. You could think of a different species or different individual, and it's colored blue to show you this. So this is not DNA cut from the same organism. It's DNA cut from two different individuals. Now, they've been cut. Now, the restriction enzyme does its work here again, and it cuts these sections off. So you see it looks a little different. So these little black restriction enzymes have cut it a second time, for making it look like this, and then making it look like this on this side. And then you see here, it says, it's hard to read here, but if you zoom in, it says recombinant DNA. So recombinant DNA sounds like recombined DNA. And that's DNA combined using these restriction enzymes to make a new strand of DNA connected perfectly. So, what is the benefit of that? Look at this. It says interpret image to the right. Summarize how the restriction enzymes help combine two species DNA. So imagine this is one species here and a species here. So what you're doing is creating a new organism by taking some of the DNA from one animal or one plant and combining it with another. So use these ideas. Bonding, which these are all molecules, right? These are complex molecules that make DNA. And for them to form new structures or to make DNA in any way, it requires bonding between those nucleotides, right? Which is this new word here. Remember in the drawings, the bars represent the parts of DNA called nucleotides. Remember, there are A's, the T's, the C's, and the G's. And then you have foreign DNA, so you can decide which one of these would be the foreign or new DNA. And then you have to realize that this is probably a gene. So the DNA is being connected in a way in order to put a new gene into an organism. So let's say this was the original organism, and then you take DNA from some other animal, and you decide what that would be, and you're adding a new gene to a new organism. And that could be a human organism, a fish, a goat, or a tree, whatever you decide. But basically summarize what you've learned about this and how restrictions or enzymes are involved in helping to combine two species DNA. But your goal is to use these words here. All right. Now, the slides show that there are 7, 8, 9, 10 slides to go. But for you, I want you to stop when you get to this slide. So we only have 2 more slides, basically. So let's look at this. It's going to be one more task to do, and it requires you to do some more writing and some compare, contrast, and it requires you to summarize your thoughts. This right here says summary of restriction enzymes. So right here, what you need to do is copy and paste what you did here. So this is the restriction enzymes. All right, so the summary you came up with right here for what is a restriction enzyme, you're going to copy that, and what I want you to do is take it and paste it into this box. So just paste it on in there. Let's see if it'll let you. Let's just make sure it's interactive. All right, so you just click in there, paste it on in, and then you go to the next one, which is right here. This was your PCR one, or polymerase chain reaction. Copy this, and then paste it down in here. All right. So you have your 2 summaries, a summary you already done and a summary right here. So you don't have to redo anything, you just copy and paste the 2 in the appropriate spots. And then what does it want us to do is to begin comparing and contrasting the 2 summaries, looking for things that are similar and different about the 2 ideas. Let's zoom in so we can see it better. All right. So it says compare, contrast, how do the 2 summaries support common and related ideas. So what do they have in common? What do they don't have in common, right? And you list them right here. So as you compare them, all right, looks like it may not let you type into it too well. So you may have to write notes right here for it. Or you can put it on paper. If you want to print this and write your answers in, that's okay too. But anyway, the idea is you need 3 things that are common to them and 3 things that the summaries have that are different. So what do they have in common? Well, here's one. They both involve humans working with DNA, right? All right, so you could put compare is DNA is involved in how humans use the DNA, right? That's a comparison, right? The contrast is that the two procedures, what humans use restrictions enzymes for and what humans use the PCR, or the polymerase chain reaction for, are different. So the use of each one is different because blank, right? So how are they similar? How are they different? And then, like it says, determine possible functions both procedures share. So how could they be used for common goals, right? So what are common needs that they both have for people? So if you're using restriction enzymes or you're using PCR, what are similar functions that they provide, right? So as a technology or using them, what are possible similar functions that that we are benefited by, or how could they work together, or so you can use both of these together as a process. Like you could, and you can kind of think about that here. So just try your best. I'm curious what you come up with. There's no right answer as long as you're trying, and then I'll give you some feedback. We can adjust it if needed. And then here's specifically what is the function of enzyme here. What is the function of PCR? So you can recognize that they have common factors. You can recognize that the two processes can provide similar functions in genetics, but they also are unique or distinct, right? So what is one thing about restriction enzymes that is different, and what's one thing about PCR that is different, right? So you're really just thinking about the two techniques. One is the polymerase chain reaction right here, and you're thinking about, again, this process of using restriction enzymes to separate DNA, right? And you're just trying to think about how can they be used together to complete tasks, or what's common about them, and then what is different. Compare, contrast, right? All right, once you fill this table in, and whether you print it or you're just writing answers down here in the notes, whatever it is, you now know how restriction enzyme and PCR are similar and different. And what you do with that information is you write a report. So the next slide, let me back up a bit so you can see a little better. It takes this table, and your goal is, let's look right here, right? So let's take the magnifying glass and zoom in. All right, it says, let me get rid of it. All right, so it says develop a model with labels, symbols, a key, action, before, after, cause and effect, procedural setup, scenario, etc. So we need to print this. So let's print. So I'm just going to recommend to print slides 7 and 8. And you can print it two-sided, or I'll help you print it. But what you're going to do is you're going to draw in here some kind of image that shows how the two are could work together, or or you can represent how each one has a cause and effect relationship. But you're doing some kind of drawing that could show potentially how they're both unique processes, but also showing how they might even be able to work together to do a common goal. And so so basically we're going to, on paper, draw with labels, right? So it says develop a model. A model in this example is a visual model, so you're going to draw what you understand about the two processes, and it's just based on what you know and understand. I'm not looking for a picture that's perfect based on like this right here. I'm not looking for this. What I'm looking for is just what you think is your best understanding of the process. So it's not getting the right answer. It's you showing what you know. It's you showing what you understand. And I have some more feedback I can provide. One second. So when you make a model, these are guidelines. And I'd pause this when you watch the video just for help. It says a model. So when you draw something, it should show. It should make you ask questions, right? So it should be about something you think is strange or maybe not understood or weird. So sometimes when you make a model, it's to show something you're confused by or what you're trying to understand better. But it also should show your best understanding of the events. Not perfect understanding, your understanding and your best effort to show your understanding. So as you're drawing this, it's your opportunity to share what you understand about it. I'm wanting original work. That's what I'm wanting. Not copied work, I want original work. It says a model should be a first draft of understanding. So the first thing you draw may not be perfect, and that's okay. The idea is that you might eventually make a second draft that changes it a little based on your new learning or your new thinking. So think about the drawing you make as not perfect, but a first draft effort that might change in the future if you want to change it. It can represent a hypothesis or claim. And you have two hypotheses. You created a hypothesis for what a restriction enzyme is, and you created a hypothesis for what polymerase chain reaction is. So you have two hypotheses you can work with if you want to use that to make your drawing or your model. It said models should show a process or action before and after, or cause and effect. So in your drawing, you want to show some kind of step. It should be some action or process. And again, the last thing is a model, when you draw it, should have words and symbols to help communicate your ideas. So you can use arrows or shapes, symbols. This is a key. So when you have a key, it says, like, what is it? So if you draw something, you need to explain somewhere that this shape or this item represents something. And so you just need a key to explain that. All right, so based on those ideas about how to do a model, use those to guide you in making an original drawing right here. And that'll be the end of the first part of Chapter 13 notes. All right, and we haven't done many models, so I understand it's going to be hard. I just want you to create this just based on your best memory of what you learned about in the earlier slides, right? And base it also on these ideas you have. So use the compare and contrast ideas to help you think about the two different things, the restriction enzymes and the polymerase chain reaction, and use these comparisons and contrasts, how they're different, how they're similar, to try to represent them in a new way, or how they could work possibly together down here. Remember, use those ideas that we just talked about for what a model should have. And I'm here to help. Good luck.